Hi everyone, this is Pavitra, working as an assistant professor in biomedical department. Today we are going to see the lecture on assistive technology. Subject code is CBM 333. Core objectives of this subject is to know the hardware requirements, various assistive devices, to understand the prosthetic and orthotic devices which is used as the stimulation tools, to know the developments in the assistive technology, to know about the recent trends in assistive technology. These are the 5 units in the assistive technology: cardiac assist device, hemodialysis, hearing aids, prosthetic and orthotic devices. And finally, we will see the recent trends in assistive technology. These are the course outcomes of the assistive technology subject. Our first outcome of this course is interrupt the various mechanical techniques that will help in assisting the heart functioning devices. Second one is describe the underlying principles of hemodialyzer machine. We will see the dialyzer unit completely in unit 2. And third one is indicate the methodologies to assess the hearing loss. We will see the hearing aids in the third unit. And fourth unit is evaluate the types of assistive technologies for mobilization, that is prosthetic and orthotic devices. And finally, we will see the TENS and biofeedback in the fifth unit. Now we will see the second unit. Unit name is hemodialysis. What is dialysis? Dialysis is a technique of removing substance from the blood through the dialysis solution using the semi permeable membrane. This is the picture of dialysis unit, and this is the dialysis process. This is the semi permeable membrane which will be thin. It contains holes of various sizes for removing the waste products from the dialysis solution. This is exactly replicates the filtering process. When it takes place in the kidney, the blood enters into the kidney, large substance will be remain, and water waste will be segregated from the blood. Why we are going for dialysis? The main purpose of dialysis is to maintain fluid, electrolyte, and acid base balance, and to remove the extraneous and exogenous toxins from the blood. Mainly, we are going for dialysis to remove the waste products in the blood. Usually, dialysis will be separated as two types. One is hemodialysis, and another one is peritoneal dialysis. Hemodialysis. What is hemo? Hemo means blood. Dialysis means passing through it, which is removes the nitrogenous waste products from the excess fluid and electrolyte from the blood by means of artificial kidney. Artificial kidney will remove all these waste products instead of our human kidney. The procedure of cleansing the blood from accumulated waste products with patient when the patient is in the stage of renal failure. For accurately ill patient who require short term treatment, they can afford hemodialysis. Nearly 90% of all dialysis patient receives hemodialysis. Hemodialysis is suitable for all the kidney failure patients. Principle of dialysis. These are the principle of dialysis. First one is diffusion, and second one is osmosis, and finally ultrafiltration. These are the different filtering techniques in the dialysis system. First we will see about the diffusion. Diffusion is the um, diffusion will be take place when the movement of solutes from an area of greater concentration to the lower concentration. It is used in the case of renal failure. It will segregate urea, creatinine, uric acid, and electrolytes move from the blood to the dialyzate with the net effect of lowering concentration in the blood. But WBC and RBC and other contents within the blood are too large to diffuse across the membrane. For that and all, we will go for another filtering method. Next one is osmosis. Osmosis is nothing but the movement of fluid from an area of lesser concentration to the greater concentration. Glucose is added to the dialysis solution and it creates the osmosis gradient, gradient across the membrane to remove the excess fluid from the blood. This is the osmosis technique. Here we will have two containers. Here we will have greater concentration and this is lesser concentration. Here osmotic gradient membrane will be created here. So solutes will be moved from lower concentration to the higher concentration bowl. Finally, we will see the ultrafiltration. Ultrafiltration is used for minute solutes in the blood. It is compared to more efficient than the other techniques to remove the extra water. It is accompanied by the negative pressure or suctioning force to the dialysis membrane. It will remove all the waste products by the pressure. Thank you.